Hello bachon, so let's read the question. The question says all of the following methods are of insertion of recombinant DNA into the host cells except. First is incubation of the bacterial cells with recombinant DNA on ice followed by heat shock and transferring to ice. Second, use of disarmed pathogen vectors. Third, PCR, and fourth is biolistics. So the key concept for this question is insertion of recombinant DNA. Recombinant DNA किसी भी organism में insert करवाया जाता है with the help of vectors. Vectors generally plasmid DNAs होते हैं. But DNA, as we know, it is hydrophilic in nature. So DNA cannot be directly taken up by the cell. तो उसको take up करवाने के लिए कुछ methods होते हैं, transformation methods. There are mainly three transformation methods. First is the heat shock method. Second is the biolistics or gene gun method. And third is microinjection. जो ये pathogen vectors होते हैं, which are used for the transfer of the recombinant DNA, they're basically disarmed. Disarmed मतलब इनका जो पैथोजेनिक जीन होता है वो उनसे निकाल दिया जाता है. So heat shock method में cells are incubated with recombinant DNA on ice. उसके बाद उनको हीट शॉक दिया जाता है 42 डिग्री सेल्सियस पे ब्रीफली. वापस से उनको रिटर्न करा जाता है आइस में. So this basically creates a heat shock which creates pores in the bacteria that take them up. Biolistics और gene gun method जो होता है, it is basically the high velocity bombardment of microparticles of gold or tungsten which are coated with the DNA. जो DNA हमको insert करना है. Third is the microinjection method. Microinjection is basically what it sounds like. जो recombinant DNA होता है, it is injected directly into the nucleus of the cell. So coming back to the question में बोला गया है, which of the following methods of insertion of the recombinant of DNA into host cells except? So incubation of the bacterial cells with recombinant DNA on ice followed by heat shock and then transferring to ice होता है. Second, use of disarmed pathogen vectors, which is also true. Third, PCR. PCR is not used for the insertion of recombinant DNA. PCR is used for the amplification of DNA. And fourth is biolistics. Yes, biolistics is also used. So the final answer for this question is option C. Hope you understood well. Best of luck.